Well as Paul and Daniel, who you've already heard from, we have Oleg Paul, who's also a member of the iGEM, Fiona's iGEM team. Actually, I wonder if somebody could just close the door. Is that okay? Thanks. Thank you. Okay, right. Who has got a good first question for the team? I'll take one here from right at the front.
Yeah. Who is regulating projects such as the internet finance ones that you mentioned to ensure that invasive species or really detrimental factors not being introduced into the environment by reckless enterprises?
Well, obviously, you hit the nail right on the head. That was my issue with it, actually, when I first came across it as a project. Kickstarter isn't regulated by a government body. Kickstarter is a separate private company. So the only people really regulating the projects that get put up on there are the company's PR. So obviously there's enough complaints and outrage from the rest of the world and the internet in general, which, to be fair, these days does happen. Then no, there's not really anyone actually regulating that project. But the project in itself came out from a university-based project originally, so it's already got some university supervisors and otherwise that are involved in turning it into a business. But in terms of making sure that it doesn't spread or anything else, that's not too clear. They've not said too much, and unless you back them, you can't find out that information. You have to pay them some money to be able to get on board. There's other sites that have seen the problem with things like Kickstarter for that. So there's one called MicroReaser, which is specifically tailored for scientific research. So instead, the idea is that you back them. You're not expecting to get any sort of like seeds or anything else back, but instead you'll be banked in papers and you'll get a little document explaining what's going on. But the truth is, with a lot of these private companies, no, there isn't anyone regulating at the moment. We're just sort of trusting that things will self-regulate in a sort of free market setup, so if it was something that would be a serious problem, then someone would do it.
But they won't, because, for example, I was working in the flood defence industry, and we were plagued by enormous growth of Himalayan balsam all along the watercourses and rivers of Britain, causing an enormous flood risk, totally overrunning native species. Who's to say that these glowing grasses won't have exactly the same effect?
To be fair, I think specifically the species they've chosen, as I said, one of them was modified roses that they have. And yeah, some of the species they've chosen are ones that are notoriously difficult to maintain without human interaction, shall we say. So as I said, it's that containment issue that I said the better way is to build in something that makes it dependent. So roses, you may know, don't always grow too well. And so.
They also sterile the plant sometimes as well, so they can't reproduce. So you just get that plant when it dies, it's dead.
But assuming they're planning on coming
with
back.
bacteria, they have things called kill switches. So they'll put a genetic component in so that it responds to a certain chemical. So when you put that chemical, it will kill the bacteria. So they do have control methods on the molecular scale to combat these kind of things. But you're right, you always run a risk of a mutation or something like that. It's just the way of minimising it.
I think currently with the fact they've got so much university backing and clout with them at the moment, I'm fairly certain there'll be enough ethicists in there that are insisting there's something to control it. I just wouldn't be able to tell you exactly what that is. You'd have to get in contact with them directly and find out.
So you said they've got enough something in there,
enough clout, enough behind them, enough support.
Cavonia, communism with power.
Okay.
Is it just my turn? I don't know. It may not be
No, it's just
saying that it can be difficult getting academics on side, is all I'm really saying. But they're never keen to part with money on things. Yeah. Any other questions?
Okay, one there, yes.
At the beginning of the talk, you talked about isolating specific genes and inserting those into bacteria. Obviously, these systems that you're talking about are extremely small. I mean, physically, how do you actually do that? You've got DNA strands, which I know that huge amounts of DNA material, specific genes. How can you isolate a specific gene?
Basically, how we do it is that around the gene that we want, there are certain DNA pieces that are called restriction sites, which certain proteins, which are enzymes, would cut the DNA at those specific pieces. So for example, in a solution, we could put our DNA and those restriction enzymes, right? And those proteins will cut out the exact piece that we want. And then through experimental method, we can isolate that piece and put it into a different DNA construct. To then, once we take that second DNA construct, put it into a bacterial cell, they are able to express only our insert of interest.
So as well, it is, you're right, because I do nanotechnology, it's my degree, and to control small things like that is really, really difficult. But what you've got to realise is there's kind of a probability of summative effect as well. You're hoping that, because you put, what you normally do when you want to modify a bacteria, you'll put something like an antibiotic-resistant gene. So this gene will stop it getting killed by a specific antibiotic, right? And maybe most of them won't get that gene inside of them. But if one does, then you can grow it on that antibiotic, and everything that doesn't have that will get killed. But the one you want, the one you've put the resistance in for, you have that. So it's kind of like a probability. You're hoping that at least one of them does it, and then you can work with that. So it is kind of like, you're getting, the more we develop, the more we're able to just edit single cells with more accuracy. But you're right, it is hard. It's like, it's like trying to build a tiny Lego house with massive hand. It's hard,
I think the only other thing to add to that is if you meant just like the actual sequencing of those genes to begin with, the answer is the Human Genome Project. Vast amounts of cash go into it. It takes a very long time. But as equipment improves and people strive towards these things, that improves as well, so it's a bit easy to do. So there's a lot of research going into that sort of area, but how we can do this faster.
Okay, question there from the gentleman in the blue jumper.
Perhaps you can tell us each how you spend your day. Do you just go on a computer, or are there machines that will put the DNA into A, B, and C, or do you have actually things that you have to do yourself to
yeah, we sort of, it's split between what we call wet work, hardware work, and then software, right? So wetware, hardware, software. Wetware is in the lab. Hardware is like actual physical things. Most of the time we don't need that. So it's only gone to like build microscopes, and otherwise software is the modelling side of things. So I spend most of my day sat at the computer screen, hating people that made the internet and other things like that because things don't work how they should, or the programming language I'm using doesn't work how it should. Whereas Oleg and Dan tend to spend more of their day in the wetware area doing well.
It is literally just basically it all boils down to clever mixing things. Science always really boils down to that. You're kind of taking two things, mixing it together. You have a theory behind it.
Basically, I can say an example. This morning, we came into the lab and I had a very nice culture of bacteria that was grown overnight. What I can do with that bacteria is that I can break it down, get the DNA out, which we did essentially. And what we did today is also get the DNA out, and we are waiting to send it off for sequencing, because there's other companies that will do it much easier. If you send your own DNA to them, they can sequence and tell you back, oh, this is the code of your DNA sequence, and this is how it is. So basically, it's partly that, partly going back and forth to the computer, because to essentially design this, you need to have primers and DNA strands, which are much easier to design on a computer. And then you would order those, you come back to the lab, and then you would do a lot of lab experiments where you isolate certain things, put them into other things, grow cell cultures, break down those cell cultures, isolate the thing you want, essentially run gels and test that. So it sort of starts with the computer, then you do a lot of experimental work, and then you kind of test it again on the computers.
So a bit of calculating melt temperatures and other things with that as well. But put it this way, if it was easy, we'd have been doing it many, many, many years before.
To be honest as well, I'm not really, I do biological science in my course, but I'm not like a biochemist. To me, it's just like cooking. You have a recipe, you chuck it all in, stir it out.
Depends on what you're working on.
You can try and make it as complicated to help you sleep at night, but that's what it is. It's shocking thing, really.
Okay, Oleg, you said you take your bacteria, you extract the DNA, and you send it off to a company?
Yeah.
You send it in the post?
Yeah.
DNA on its own is harmless. It's not, it has no effect.
Basically what we do is get a bacterial culture. We would use a centrifuge to isolate the cells, and then use a certain chemical to break down those cells to elute the DNA that we want. Then we would concentrate DNA, and essentially after that we can send it off to a company that will then sequence it much cheaper for us than if we bought our own, the University of Leeds, if they bought their own sequencer, for example.
You literally send it off in a little sample bottle.
It's very well sealed. There is that. I mean, It's still a viable
Tends to,
product,
yeah.
so it tends to get sealed up, but I mean, it's not likely to break. It's not exactly a glass vial. And in terms of getting the stuff out, you may have seen on Dara Breen's science show on BBC, at one point they did a demonstration where they used vodka to separate the DNA out and things like that, so it's very easy to make sure you just get the DNA.
I've actually done that.
Yeah.
Typical.
And
Yeah,
as we said,
typical Russian,
DNA on itself, DNA on its own doesn't really do anything. It needs a lot of enzymes to do stuff, so it's pretty safe.
Okay, right. Question there from the front row.
On the beginning, on your screen, before you started talking, you showed the process of producing human insulin by bacteria.
Yeah.
I sort of gathered from that you produce the insulin to then give to humans.
Mhm.
Have you not thought, or have you thought that it's not feasible, instead of feeding them into a bacteria elsewhere, to feed them into actually make it so you could orally or inject them into people to produce that last stage, so they're not continually taking a medicine, so to speak,
Right.
but have
That is actually an area called gene therapy of science. It's a bit different, but what you've got to realise is, say the genetic code is a recipe to make that insulin. Now, if you have diabetes, you don't have that proper recipe. Now, all your cells have that wrong recipe in them. So you've got to change every single, well, or rather the cells that make the insulin, you've got to change every single one of them to do that. So it's a bit of a different branch of science, but they have something called gene therapy, which is also a very controversial and a very new part of science, where they take a virus and they put the genes they want in that virus. And basically it's how a virus works. It will infect your cells and change the genetic components. But it's very dangerous as well. You've got to take a lot more control on that, because you don't want to start creating super viruses that go around changing your body. But you'd have to change every single cell, pretty much. So it's quite a lot more difficult, because we're doing it from the start. So we create the first cell, the mother cell, and then grow it up. But when you want to change cells that already existed in a massive state, it's a lot more difficult.
right.
The other thing as well is to bear in mind the differences between different diseases. So for diabetes and so on, you've got two different types. You have those where, yes, the person just isn't producing insulin when they need to, and in other cases it's that they can't regulate that production. So injections of insulin are better because they can time it exactly and go, I know how much I've eaten, I know when they need to have the insulin. My cells can't work it out, but I can, so I inject it. So they need to have a liquid insulin effectively. And the point is, it's a lot more efficient to produce that from bacterial cells that can just eke it out everywhere and just strain it off effectively. It's the equivalent of putting a bit of muslin cloth under a cheese and straining it and going, oh yes, the whey from this is what I want. If you see what I mean.
And also, people are getting annoyed at us modifying bacteria if we start genetically modifying humans.
That might not go down as
It's
well.
much more difficult to modify human cells than bacterial cells. And bacterial cells grow very quickly overnight, so it's much easier to get the product that you want, basically.
Economics.
Okay, we've got a question from over one side.
Just a comment, you'd be pleased to know that the first clinical trials of gene therapy for heart failure just started. First patient was recruited only last week ago, maybe last week, so things are happening. As you say, it's a long road.
It tends to be something that's disease dependent though.
Potential you could cure, if you could get gene therapy to work properly, you could cure thousands of diseases, like pretty much like that, if you could actually get a viable way to get it into the cell you wanted to. But it's a big plot, though, so.
Okay, we have a question from the cheap seats.
Can I ask you how sensitive your little microbeaver is? Because, I mean, if I understand it correctly, to get a fluorescent signal,
Don't speak so loudly, it's quite simple.
you need to have bacteria interacting.
Do you want to know how sensitive it is currently, or how
Yeah,
sensitive
sorry,
it will be?
sorry, just for some people at the front, do you mind repeating
Sorry.
the question?
Right, so your question is how sensitive is microbeagle to whatever was triggering it? Do you mean currently, or do you mean in time to come?
Well, what I'm asking is really, do you think it's going to have to be very sensitive? Because if you're talking about some of the pathogens that cause diarrhea, like salmonella,
Currently,
you're talking about an effective dose of sort of 5 bacteria.
currently it's oversensitive.
Currently, due to the pathway we're modifying, it kind of triggers off a lot, so it's quite oversensitive. But what you're saying is you need some sort of amplification system. And I think you remember I was talking about the biofilms and the quorum sensing, and the bacteria talk to each other. So scientists have actually started to manipulate this fact, and they use this. So they use the bacteria against themselves. So say one cell detects the pathogen. Then you could have a genetic marker, so when it detects the pathogen, it releases all these chemicals, the chemical language I was talking about. So as soon as it detects it, it's like, hey guys, pathogen, cool. Right? And then it will obviously amplify up and all of them will have a reaction. So that way you can make it as sensitive as you want it to.
To be fair, at this point in time, we don't have a, that's one of the problems that we have, is that we're looking for a threshold where, for example, if you have a bucket of water and you put our sensor into it and say maybe there's a few molecules of cholera, that won't necessarily make you sick if you ingest that water, but it will set off our, our sensor. So right now at this point, it's more for further research, because in this span of the summer and of our project, we weren't able to kind of incorporate that into our research. But for future projects, it's definitely one thing to watch out for, is to make a threshold where to say, okay, above this certain concentration of lethal pathogen, this sensor will start working. So that's future work, essentially.
Okay, question here.
Following on from that, suppose you have a bucket of water and you're testing it for cholera bacteria, and it doesn't react, it doesn't glow, so you think, good, I can drink this. I suppose you've already got cholera in your system. Do you then glow?
The idea is not to drink microbeagle. The idea is normally, for whatever water you're going to sample, you obviously take a small amount out and put it in to a separate tube. We wouldn't say put it, no, you don't really want to, I mean, he is mostly harmless, but still don't really want to drink it.
It's a protein. The fluorescence is just a protein, so you could, in theory,
you could if you want.
They've actually,
we make
I don't particularly, they put this in cats. They have glow-in-the-dark pets, and goldfish and stuff.
There is a serious reason.
It's safe.
You should be organising these guys' end-of-term party, I think.
Yeah. I don't know if I can take it.
But no, the idea would be to take the sample out, and at the same time we've then seen with things like TB, for example, you could do the same thing, like you can take a blood sample and do it, but you don't have to obviously put it into the person.
Yeah.
That's fair enough though.
Okay, yes, Phil.
So in terms of sort of rolling out this beagle idea, I know you're very much at the conceptual stage here, but on the ground, when you want people to use it, how do you see people using it on a daily basis? So they've got a bucket of water.
Probably we'd start off with whoever's using it. To be fair, it's probably not ideal for absolute bottom rung developing countries. You'd probably want to have a centralised bank somewhere that can at least store the bacteria in the meantime and grow them happily. The idea is that someone that go out into the field and carry it with them. The point is that a lot of the time with field equipment you need electricity, you need all sorts of other things. It's very difficult to have good field equipment that's sensitive enough and so on. So the idea instead would be that whoever's going out to test the water and test like a sanitation system or something can go out with these very cheap little vials, take a sample, pop it. So they just need to take a briefcase filled with these things that are stable for a good day or two in that case. And if you try getting through Kenya, for example, it's going to be difficult to get to certain areas, and it's probably a lot easier to go with just the case of this stuff. So that's where we're seeing it be more likely used. At the same time, if you've got conditions where you could keep it safely enough and otherwise use it on a daily basis, so even in large cities, if people could have it in their own homes, like you could potentially keep it in a fridge in the same way people grow their own yeast for bread these days. You could potentially do it that way, although again we come into the sort of containment side of things.
So is there a sort of frequency by which you'd have to use it? So if you tested today, would you be able to say for a week that water is fine? You have to test it
That depends.
It's at that point in time.
It's at that point in time, really, but that depends on the system you're testing. If you're testing a river, then obviously no. Within the next 30 seconds, you don't know what's in there after the next inch of water you take up. If you're testing a national sanitation system, like we have water pipes and everything else, if you're testing that sort of thing within Africa or even just southern Italy, for example, where there's a lot of the time you can't really drink water straight out of taps, for example, it's not properly cleaned. For things like that, it's actually very good for testing on it, because obviously you'd have a much longer period before you can't be sure that the reading is accurate anymore. And that's kind of already covered by engineering in these fields anyway. So it depends on the system that someone's using. But I'd imagine, yes, most cases it'd be good with this.
Moving away from the developing world as well, you can also combine this technology with electronic technology. You could have like a meter, a fluorescence meter, for example, that reads the exact fluorescence and actually takes a quantitative amount. So the really cool thing about technology at the moment is biological technology and classical computing and electronics is really converging. So those kind of biosensors would be even more accurate, but obviously then you need electricity and a lot more things which might not be suited for the developing world.
So have you thought about who you partner with to roll out technology.
We've been
Not
trying.
Monsanto. Not Monsanto.
Yeah. I mean, currently, as we said, we're still in the development stage of things, so we try to get hold of things like Yorkshire Water and other water companies within the UK first, just to even see if they wanted to make use of it for their systems. Problem is, even getting in contact with the people there is difficult. It's surprisingly difficult to get in contact with them.
Rejection is never good to have.
More than likely, if we had a system that we knew worked perfectly, because that's the other thing, we don't really want to approach charities until we've got something that definitely works and we can give them absolute costings and things like that. But that would potentially be the way to go with it. At the same time, it's research work, so it doesn't always pan out. There's stops, there's starts, there's things that prevent it.
Okay, question from the back row.
Are you just looking at this, whether you testing pathogens in water, or would this technology be used for testing biological samples to diagnose disease as well?
It could be used for a lot of things. One of the things I suggested originally actually was maybe even looking at the idea of testing for TB. My girlfriend's here, she's going to be embarrassed with me now for saying this. Her father's a vet in Northumberland, and he'd already said before now that when going around to test for TB in cattle, for example, the problem is that the current test you use is the Heath test. Now, you may know about that. It's the one where you basically jab them, it infects them with a certain strain of TB-related bacteria, and it'll either show up or it won't. The problem is, once you've done that test, you can't reuse it. So if you find even just one of the herd has TB, you have to kill the whole herd. Ideally with ours, what you could then do is instead go out, take a blood sample, and in the field, pop it into one of these things and go, okay, oh yes, this cow's got TB, that has to go and be slaughtered. But you can test the others and go, oh, these aren't testing positive for it. That could just be the sampling that's gone wrong or whatever else. But it means you can go, okay, we'll leave it, I'll come back in a week, test these ones again, and we'll see if the rest of the herd's got them. It's not going to cost any more than it did before because our system's very cheap to do. But at the same time, you could potentially then not kill all the cattle. You could actually have lots of cattle that are there. And that's a big saving for vets and actually a big saving for the farmers on that front. But then if we could do that with cattle, we could do it with humans, we could do it with any other biological sample. So the trick really is in finding what we call the binding moiety. So the thing we're using was the silica binding region. So when we've got the silica bead, we have something that binds onto that. You can change that out. There's a protein we're using to express that on the surface in the first place. It's very easy to change out what you have expressed there instead. So we could quite easily change it to something else. It's whatever you wanted to detect. Be it you wanted to detect, say, arsenic in water, or if you wanted to detect other pathogens. So potentially, yes, whatever you want to detect in a solution of some sort.
You can also take it further. The beauty of this system is it's modular, and what I mean by that is you can change parts out. It really is like computer programming. You can modify it slightly. So you could do it for heavy metals, like poisonous metals in the system. You could do it for a range of things, and you can coat it. You can coat these bacteria on the surface, combine it with other stuff. So, like for example, I've literally just made this up off the top of my head. You could have a toilet which has these bacteria coating, and it will detect toxins in your urine, for example, and fluoresce if so. So you could have early detection systems like that implemented.
Use it to let people know they've been drinking too much and shouldn't drive.
Moving on, right, question over there.
I was just wondering, what you're talking about here is the technology for identifying these bacteria, but does it not lend itself as well to killing the bacteria, so that you, rather than just telling you you've got a lot, it kills them?
So, as Dan said, it's very modular. So all we do is we change out this little glowing tail for, say, a big nasty spike of some sort and just have it kill the thing. Effectively, what you're asking there is, can we make it into a blood cell, like a white blood cell that you have in your own immune system, which is kind of cool. But potentially, yes, you could do that with the bacteria, so you can make it something that wants to attack
That's what's
these
cool.
things.
You can take the part that senses from all the stuff we developed, and you can combine it with a human white blood cell and the mechanisms this uses. So you can literally take, like, this is like a collage, you take bits together, you put the parts to get what you want. But yeah, it's completely feasible to do something like that. Also, what I was talking about quorum sensing is kind of more a sneaky, tactical way to take them out. But you can trick the bacteria, because like I was saying with the chemical language, but there's actually different languages for different bacterial species. So some of them are bilingual, for example, and if you can work out sort of the Rosetta Stone of these languages, you can trick them into killing themselves or doing something stupid that will make them die. So there's loads of ways you can go about it, and it's really, you need someone to come with a good imagination and work it all out. Sky's the limit.
Okay, we have a question there.
Right. I was interested by this fluorescent stuff, particularly in terms of power.
Right.
Does it just keep glowing forever, or?
Well, the protein itself has a half-life. I mean,
yeah, essentially, on the DNA itself, there's a promoter. So, for example, if in the system that we have, once our biosystem binds to the silica, the promoter will be activated and the green fluorescent protein will be produced. Now, this protein will take, well, it's a bit inconsistent.
About 8 to 20 minutes to actually produce.
Exactly.
the one that we're
It
using.
will take a bit of time to for the cell to produce this protein and then for it to fold. Once it has folded, it makes a special ring on the inside, which will then fluoresce green when it comes, in relation to light. So, for example, this protein does have a half-life where after a while it will degrade. And you can see that it degrades if you leave, for example, the bacteria in the fridge without, any stress, or in our case, any binding affecting onto it. So, I mean, in your question, for example, yeah, it does degrade after a while. Therefore, in our sample, it's as soon as it binds, you will, after maybe half an hour, you will definitely see a reaction.
Another thing you've got to consider as well, you've got to keep the organism alive as well, you've got to keep it feeding. So it will be produced and it will decompose, but the rate of decomposition has to be balanced out by the rate of production. So you need to have a net amount being produced.
Essentially, if you stick the sample into the fridge and the bacteria not, there's no metabolism going on, or there is metabolism, but they're quite dormant, not much of the protein will be produced, therefore not much of the green color will be produced, even though the binding may or may not be happening. So essentially, you would want the bacteria to be in a happy state to be producing the protein, because I mean we are using live systems. It's not
plants as well. So plants are a bit more easier.
It.
Like you can keep a plant happier. You can see it easily if it's happy. It kind of droops to the side.
I'm just wondering in terms of developing sort of like producing like power.
There are people that are working on other
Yeah.
projects doing that. They aren't necessarily using the fluorescent protein. They're using.
That's more
sort of
nanotechnology. It's kind of combining technology.
yeah.
But it's like you kind of have to, if you want to create electrical power, remember, our binary systems, our computers are man-made things. So nature isn't compatible, if you like, with it naturally. So you've got to work out a way of developing energy and converting it into an electric current. And I would love to tell you how to do that, but I wouldn't be here right now.
The other issue with that as well is,
to be honest,
biological systems are notoriously inefficient for energy usage, shall we say, because they have to live, like about 80% of the energy, it's going to be used for growth. So for energy production, not necessarily ideal unless it's doing something specific. So there is an iGEM team that's working on making a hydrocell, so a hydrogen water battery setup, so it's very green energy. But to help catalyse it, they're basically using the bacteria to do the processes they need, because there's quite a lot of complex chemical steps, but the bacteria can do it very rapidly, because it's effectively the same processes we use in our body to relinquish energy from what we eat. So it's sort of making a microsized biofilter sort of thing.
That to me as well, what you're suggesting is like the pinnacle of technology, when you integrate electronics with humans, and like you can actually fully, it's basically human augmentation it's called, and there's a lot of, it's completely theoretical, this field of science at the moment, but there's a lot of issues. Like one day you could be able to just, put your fingers into your phone and charge it, or, send people messages through Wi-Fi, but that's in the future. But definitely, it's not impossible. It's definitely, the technology definitely could be developed. It's just how it's the way.
Okay, do we have any other questions? Another one at the back there.
Can I just ask, how stable is culture if you're in the middle of Kenya and you've got just a incubator or a fridge or whatever?
That's a good question.
And you've got your culture. How easy is it to propagate that? Does it degenerate after a few generations? Or?
It depends on what conditions the culture was brought in.
So Kenya, for example. So it's quite a hot, humid environment, and it could cause denaturing, it could cause damage to bacteria. But you want to take, firstly you've got to look at, aside from the bacteria, you've got to look at preventative methods, so storing it in a suitable container, making sure they have nutrients. But if they have nutrients and they're not too extreme environment, then it should be pretty easy to store them.
It doesn't degrade as it
Do you mean the actual genetic modifications we've made?
Yeah.
Not really any more so than you'd expect with bacteria, I'm afraid. It's DNA, that's the point. It is notoriously bad at holding information, that's why we're all here. But at the same time, the bacteria are actually pretty good at it. They have other ways of dealing
DNA
with
is
it.
not bad at all.
This is often why we often put in the need for antibiotics in some ways, is by having an antibiotic resistance in there, you can make sure that those that mutate away kill off more easily.
Essentially, whatever inserts and whatever proteins we put into the genes of the bacteria for them to produce later on, they will stay there. The main question is to keep that bacteria as a whole alive.
Yeah.
And we can do things like isolate them into glycerol stalks, where we essentially kind of freeze bacteria in a state where they can be defrosted, then put into a nice, happy environment where they are able to grow with nutrients, and overnight they will make a very nice bacterial culture. From then on, you can use that bacteria as a sensor. However, if the storage temperature, exceeds whatever it's supposed to be, the bacteria will be lost. So again, we're dealing with live systems, and you have to be very careful to preserve them.
So basically, the genetic parts are really stable, but the hosts themselves are more vulnerable.
Certainly within the space of a day, they're still functional. I mean, I've done some experiments with AFMs, so atomic force microscopes. They're very good at measuring all sorts of things. And there was an experiment I was trying to do with it, which meant I needed to shift samples back and forth, and I needed to make sure they're on some slides, prepped early, not stressed, and all this sort of thing. You've got to make them relax, a little massage here and there. It's a lot of fussing and things. But they were basically, they'd spend about 1 to 2 days on these slides anyway in just the fridge, which isn't exactly an ideal condition, but enough to keep them fairly dormant. And they'd be quite happy to be there. And by the time we got them onto the AFM, if we left them on there for more than an hour, we actually saw them growing actively. Because we have a fluorescence attachment on the scope as well. So you can see them growing, because every time they grow, you get this very long, bright spot on the scope that's like, oh dear God, that it's producing a huge number of bacteria, and they just split off, and then there's more, and there's more, and there's more. So they're actually a little more resilient than you might think, but yeah, they're not invulnerable. Anything else?
Okay. Right, well look, that's good. I think you've answered everything that could possibly have been asked. So, yeah, okay, well that's great. Thanks very much, guys.
Cheers.